after few days, after, right? So few days, like 15 to 20 days or 1 month time here, right? So after the few days, when he observed, right? So when he observed that the bacteria is, right, in this particular culture, right, in the petri dish, generally that culture is done in the petri dish. Actually, the biologist will, that means the biologist people will know about that petri dish, right? So here the microbiologist, they use the petri dish to develop the uh, right, to develop that particular culture, bacterial culture or the virus culture, whatever, right? So in this petri dish here, right, so the bacteria is developed here, right? The bacteria is developed here, so it is consists of bacteria here. And here in this particular petri dish, some fungus also produced here, right? So in, okay, so in this particular layer part, right? So some fungus is produced here. Okay, fungus is produced, and where that fungus is produced, at that place the bacteria are, right, they are killed, right? So no bacteria is developed at that particular place. So then he got the conclusion, right? He come into the conclusion that, so that particular fungus which is developed, right, in that petri dish, so it may consist of biological activity. It may consist of pharmacological activity which is acting against that bacteria here. That's why the bacteria is not developing at that particular place here. Okay. So, and later it is found, right? Later he's coined that particular uh, active compound, right? Active compound consists in the fungus is the penicillin. Okay. So that conclusion made by the Alexander Fleming, and further then after that, so some three scientists like Florey and some other scientists are, they are synthesized that penicillin here. So like that. So here you see that particular uh, penicillin is a fermentation, okay, drug here. It is produced by fermentation method here. Okay, during fermentation it was produced. Okay, fine. You see the general equation, the general method to produce the products here is by fermentation technology, where the microorganism is taken, and to this microorganism some the substrate here. You can add substrate, so that's it's the substrate. It may be the sugar or any other things here. Right? So here in most cases we will use carbohydrates here, sugars. Okay. And along with this some nutrients, right? So some nutrients are used because these nutrients are consumed by this microorganism, okay, for healthy, right, for healthy the growth of this microorganism here, the some nutrients are required, okay, like what is that, so phosphates and ammonia, right, ammonia, phosphates, so all these nutrients which is useful to maintain the, right, that particular uh, fermentation method active here. Okay. And then after few reasonable time, so you will find some products here. Okay, you will find some products, and these products depending on that okay, depending on the scenario of that fermentation procedure, right? So we will get the four kinds of products are obtained here. Okay, so here you see that the variety, the different types of products which are obtained by this fermentation method. During this one, you can see that the one is a microbe themselves, right? Okay, that means here you see sometimes the microbes, right? So they are okay. The microbes also, they will produce products here. They will means like they will act as a products. Yeah. You see the yeast. Okay, yeast. So here in the bakeries, you see, right? So in the bakeries here, bakery to you have, everybody will have the experience the bakery products here. So in the bakery, when you prepare the bread here, right? Okay. So the bread preparation, it is a fermentation technique here. So to produce this bread here, the yeast, okay, yeast here, okay, and the curd also, right? The curd. So preparation of the curd, this is also fermentation method here. Curd preparation, this is also fermentation method here. So like that, you have variety of okay methods are there in right the regular life. You can see variety of products which are formed by the fermentation method. Okay, right. So here the microbes themselves they are acting as that right. So the act means fermentation products here like yeast here and the single cell protein. So right, the single cell protein. So it is having some right single cell protein more energy here. Single cell protein here, right? SCP, SCP here, single cell protein. Okay, and here you the microbial enzymes. Okay, here the polymers are the large molecules here, and the enzymes and the polysaccharides. So these are so products here, and the microbial metabolites, right? Microbial metabolites here. Means here by this fermentation method, we are producing the products like right. You can produce a yeast, right? You can produce a yeast compounds here by fermentation method and single cell proteins and some microbial enzymes. Yeah, there's some enzymes which are useful to produce some of the useful products here, right? Like right, some enzymes and polysaccharides and microbial metabolites here. Microbial metabolites, right? So here the microbial metabolites. So we know that what are the metabolites here. So the metabolites which are the means the products which are produced by the metabolic process here, right? Like we know that you have an idea about the metabolism here. So where it involves a catabolism and anabolism, isn't it? So where one is a constructive method and another is a destructive method here. Okay, constructive method and the destructive method combining it is called the metabolism. So here the during that procedure, during that metabolic process, the products are obtained, the products are formed. They are also useful materials here. And in that particular metabolic, right, the microbial metabolites, we have two types are there, right? The in the metabolic metabolites, right? In the metabolites, there are two types here: the primary metabolites and the secondary metabolites here. So generally, what are the primary metabolites here? So the primary metabolites which are useful for the survival of the living organism, means they are essential for the growth. Okay, the primary metabolites here. Okay, so here the primary metabolites like amino acids, vitamins, right? Nucleotides here. Okay, enzymes, proteins. Okay, so all these are the amino acids. All these are the primary metabolites. Okay, and they are essential for this for the growth, right? They are essential for the growth and they are essential for the survival of the living organism. So without primary metabolites, the living organism cannot be survive here. Okay, because you say you can means we can't assume without DNA, is it? Because the DNA is a means in the nucleic acids, right? The nucleotides are the nucleic acids. So like in that DNA and RNA. Okay, so here the DNA and RNA, these are essential uh, biomolecules here, right? So for the life process here. So without DNA, you cannot, right? You cannot assume the life here because the DNA is the primary material, right? The primary material for the protein synthesis. Okay, the primary function of this DNA is it involves in the protein synthesis by the translation process here. Okay, the DNA. So it involves the translation and transcription. Okay, it involves in the translation and transcription process here, where the translation is a protein synthesis, transcription is a mRNA synthesis. So here the translation by translation method, where the proteins is produced here, and that proteins are essential for the energy. Okay, without proteins, the energy will not be produced here. Okay, so that energy is not released here, and if there is no energy, then no biological functions are takes place here, because to carry out any functions, to carry out any biological function, the energy is required here. Okay, so that energy is produced by your particular DNA. That's why it is an essential, right? It is an essential material for the life process here. That's why you called it is a primary metabolites. And the second one is the secondary metabolites. Okay, the secondary metabolites. Generally, these are not right. These are not useful for the producers here, and they are not. These are not non-essential for the growth here, right? So without secondary metabolites, the living organ, it will be all the functions will be takes place here, like all the metabolic process like respiration, circulation, excretion, okay, digestion, and reproduction, whatever, all the functions, all the metabolic functions, everything will be uh, carried out here without this secondary metabolite. They won't have any importance in that, right? They won't have any importance in the life process here. But the primary metabolites having the importance in the life process here. Okay. So here in the secondary metabolites, you have variety of products like okay, okay, antibiotics here. Okay, and drugs, pigments, poisons, and perfumes, and alkaloids, terpenoids in the natural products, right? In the natural products, the secondary metabolites are alkaloids, terpenoids, steroids, carotenoids. Okay, all these comes under your secondary metabolites here. And along with this, in the artificially prepared, these are the antibiotics here, the drugs, okay, pigments and poisons and perfumes. So all these are the secondary metabolites here. Okay. Okay, and here you see that the secondary metabolites are these are downstream products of microbial synthesis, okay? And they are synthesized from one or more primary metabolites. Okay, so like because these secondary metabolites also produced by these secondary metabolites. These are essential here. To produce this, the primary metabolites are required, but the secondary metabolites, they won't have any importance in the life process here. Okay. Next, here the products of microbial biotransformation are the precursor fermentations, right? Yeah, the precursor. So what is a precursor generally? So in the means here the precursor means the starting material here. Precursor nothing but okay, the starting material here. So in your right, starting material. Precursor means the starting material. Okay, so in the retrosynthesis, right? You will have that terminology in your retrosynthesis, synthetic strategies. Right. So here you see the precursor, the smallest molecule or the smallest starting material, which is involves to give the final compound, final product here. Okay. So like you see the starting material gives a product. Okay. Target molecule here. Right. Starting material here, other starting material, it is giving the target molecule here. Right. Other reverse means because in the retrosynthesis we will take the target molecule here. Okay, and we'll do the what is that, some transformations or the some logical, right, the logical disconnection, and we'll get that starting material here. Yes, sir. Right, so so generally this is called a precursor, right? So this is retrosynthesis. Yeah, retrosynthesis symbol is this one, and by taking this starting material again we will produce this target molecule. So that is the retrosynthesis process here. Okay. So like where the transformation of the precursors by the fermentation technique or the method here. So where this the foreign substrate, okay, this is the foreign substrate. So it foreign substrate nothing but any molecule compound here, right? The molecules, and it is going to uh, convert into products in the presence of your microorganism. Okay. So here the one example is given, like you see, right? Here the testosterone here, right? Testosterone, sorry, progesterone, right? So progesterone, which is the female, right, genital organ, means hormone here, right? The female sex hormone here. Here this progesterone, right? So where it is going to convert into the what is that cortisone, okay? It is converting into the cortisone in the presence of the Rhizophus nigricans. Rhizophus nigricans, it is right. So it is Rhizophus. It is bacteria. Here. It is the microbe. It is a microbe, bacteria microbe. Here, right. So in this, in the presence of this Rhizophus, where right. So this particular progesterone is going to convert into the first in the first step where it is going to convert into lemon hydroxyprogesterone here. Right. So what is happening here? Where this yeah, the introduction of the hydroxy group here. Right. So at 11th position, the hydroxy group is going to introduce here. Okay. And after in some other steps, it is going to convert into cortisone. Right. So this is a steroid. Okay. The other steroid is going to form here. Cortisone steroid is formed. So this conversion is right takes place in the presence of Rhizophus bacteria. Right. Okay. And here is a sub the synthesis of this particular cortisol artificially, right? So this is the biosynthesis. Okay, this is the biosynthesis of the cortisol here. This is the biosynthesis here, right? We call this the biosynthesis. Means with help of the enzymes or the bacteria, but the living organisms. Okay, so in the presence of the living organism. So this is biosynthesis. But same cortisol, right? The same cortisol is produced artificially in the laboratory by involving the 31 steps here, right? Where it is produced involves the 31 steps, right? So you see, okay. And this particular, you see that process, okay. So what are the right? So what are the ingredients or the materials are involved in this particular process? You see the biocatalytic process here. So in that where involving all these other right, all these other materials, right? All these other right. So materials which are going to used in this fermentation technology, like the growing cell fermentation method. These are methods, right? You see, and the multi-step reactions here, and here the here you see all these other so growing cell fermentation. Here this it is a multi-step reaction here, and generally this is not takes place outside of the cell, right? So it is in vivo only. Only in vivo, by in vivo method, you can produce this uh, biocatalytic reactions here, right? And by taking the cell-free enzymes and whole cells here, it is enzymatic process, right? So the difference, right? The difference between the right laboratory synthesis, lab synthesis here, and the biosynthesis. right? The major difference is where, okay, the major difference of this, it is multi-steps here, multi-step reaction here. It is single-step reaction, right? Like here, your rhizopus, okay, the progesterone, which is converting into cortisone, right? So where that single step with help of the like microbes here. But in the biosynthesis, lab synthesis, it is multi-step. So like if you go with the artificial synthesis, lab synthesis, it involves 31 steps here. Okay? That's why it is like the multi-step synthesis and is it won't take place outside of the living cell here. Okay? The advantages, there are some advantages are there and some disadvantages are there. And yeah, so now you see the history of the fermentation. So where means here this fermentation method technique, right? When it was taken up and how it is started and what are the right events and what are the right the events and what are the what is that called the struggle, right? And the sequential development, right? That how it is developed here. Uh, so you can see that. Okay. So like here in 8,000 years, okay. So here the microbes are produced food and drink before they was observed to a primitive microscope in in the 70s. Generally here, this fermentation technique is developed in the observed in the 17th century itself only, right? So now it is 22nd century, it is 22nd century, but in the 17th century itself, it is observed and it is some work, research work is done on this fermentation method. And who was first observed and who was, okay, who carried out this fermentation is Anthony von Leeuwenhoek, is a microbiologist here, Leeuwenhoek, right? And later right? Later in the 18th century, in the 17th century, Anthony von Leeuwenhoek, microbiologist, he was observed some and he was done some research work on that particular fermentation techniques. And later that the another biologist, microbiologist in 1857, the 18th century, right? So Louis Pasteur, famous microbiologist, scientist here. So he was right. He showed the alcoholic fermentation, which is caused by yeast, right? Yeast microbe here, and he has also postulated that microorganisms are the responsible for infectious diseases, and launched the germ theory of disease. Okay, germ theory of disease here. Right. So in the germs are the microbes. Right. Some microbes are the germs are useful and some are the harmful to us here. Right. So that postulate, right? He was postulated that these germs are okay. They are harmful and they are causing the diseases here. Right? And in 1880, where the lactic acid, right, as I told in the beginning, the lactic acid is the first, right, first optically active compound which is obtained industrially, which is the obtained industrially by fermentation technique here. Right? Lactic acid, it is optically active actually. Right? Lactic acid, it is optically active. So you see the structure of the lactic acid. Right? So first the lactic acid, which is produced right, industrially in 1880. And later in the 1914, during that first war, yeah, right? So due to the shortage of stone, right? So in place of a stone, here they are prepared some other material like right some other material by this fermentation technique, because the shortage of due to the shortage of this stone raw material here. And this stone is used in the smokeless powder, right? So means which is uh, right. So it is used in the war here, right? The First World War, they are used that particular right cordite. Okay, the cordite, it is explosive here. And to prepare this cordites, where the stone is the primary material, primary starting material for this synthesis here. But the stone, there is no sufficient sources, sufficient raw material of that stone here. That's why where that it was developed, right, the isoform means the right, the another isoform of this stone is produced by fermentation technique like stone butanol, right? Stone butanol is developed by this fermentation method by using these microbes like what is that bacterium, uh, clostridium, what is it, butylium, right? Bacterium, clostridium acetobutylium. So this is the microbe which is producing this stone butanol, stone butanol, which is a raw material. Stone butanol is a raw material for this right cardite here, right? So this is the raw material for this synthesis of cardite here. Cardite, it is used in the right. It is explosive here. It is explosive. Okay, explosive. Okay. Generally, it is prepared by this stone here actually, but due to shortage of the, they are right replace this stone by stone butanol, right, and it is prepared by fermentation technique and later. So all these are the sequential, right, the development of the fermentation technique, uh, right. So during the particular 17th century to progression, right, how the progress of this fermentation is takes place and what are the efforts done and how the means how the variety of compounds are produced by this fermentation technique. So the sequence of this development of fermentation technique you see, right? So the first one, right, the very first fermentation product is the what is it? Citric acid, isn't it? Sorry, lactic acid, not citric acid. The lactic acid, they have prepared the lactic acid, and later they produced the acetone butanol. And after that's in the 19th century, in the 1923, where the citric acid are produced by right. So produced by the what is that? Fermentation process from sugars here, right? The lactic acid. So here by using this Aspergillus niger, right? Aspergillus niger. By using this Aspergillus. This is Aspergillus. It is a right. Aspergillus. It is a bacteria, I think, right? So by using this, they are produce this citric acid here. And here you have right, so like citric acid and lactic acid. These are having a variety of applications here in the industrial, right? Industrially, they have citric acid. It is having variety of applications and it is used in the variety of products here. Citric acid, right? You can in variety of products like in the what is that in the food items and some what is that the drugs also, right? Or in the syrups also, the citric acid is using. Okay. Yeah, and in 1928, where the Alexander Fleming, he was made a chance observation of the uh, producing of right, chance observation means with that penicillin. He was invented the penicillin by chance. What is a trial and error method actually? So we call this right. So in 1920, 19th century, 1928 was this penicillin here, right? So penicillin, So here the penicillin, and generally we call it is a chance observation. Chance observation means here the trial and error, yeah, right? The trial and error method. So why it is because, and we call it, right, the penicillin. It is a serendipity of drug. Right? This is the best example for this serendipity of drug here. Very important terminology. Serendipity of drug, nothing but unexpectedly invention of the drug is the serendipity of drug. In this category, the serendipity of drug, the penicillin is the best example for this serendipity of drug because the penicillin is invented unexpectedly, right? Because the Alexander Fleming in 1928, generally he kept a the bacteria culture here, but unexpectedly he was found some fungus is formed in that culture, and it is killing the bacteria. That's why that at a particular specific part, the bacteria are not developed here, and that not development of this bacteria is due to formation of this fungus, and that fungus is having some right. That fungus is having some kind of pharmacological activity or the biological activity, right? That's why it is killing that active compound in that fungus is killing that bacteria. And later he was find and he was invented find that it is the penicillin here. Right? So Aspergillus, what is it? Staphylococcus aureus. It is a gram-negative bacteria. Right? It is a gram-negative bacteria. Okay, so that's why he called it is a serendipity of nature, unexpectedly invention of it. And he was only the Fleming. He was find that the active compound is there, but he was not isolated that active compound actually. Okay, but later in 1940, these three scientists like Flory, Chain, right? So these scientists they are prepared the penicillin in the laboratory also. They isolated and they will prepare the penicillin in the laboratory. Okay, in this way, right? So by step by step here, this fermentation technology is going to develop here. Uh, okay, and later here, this fermentation technique also applied to produce amino acids, vitamins, enzymes, and hormones here. Nowadays, right? So nowadays we are applying this fermentation technique in all field here, not only the right in the bakery items and right what is that alcohol preparation of alcohol items like wines. Okay, but it is applying for preparation of variety of uh, fields like in the drug, right? In the drug industry also they are preparing here, applying this technique here. Okay, so it is spread over all the technique means fields here, and we are going to produce these amino acids, vitamins, enzymes, and hormones. All these, right? All these materials are prepared by fermentation technique here. And yeah, and one more thing is it is going to uh, involve in the recombinant DNA technology also. Okay, so now we'll see that. Okay, so in the industrially, right? Industrially, the fermentation technology, right? Industrially, the fermentation technology involves variety of, right? Means they requires the variety of materials, and they involves the variety of stages and right process here. And what are the requirements in the industry to carried out this fermentation technique here, and in what kind of techniques this fermentation is utilizing here, and you will see. here you see the catalyst means the basic requirements for this fermentation technique in the industrially here you see the catalyst. Generally the catalyst is a very essential material because to convert the product, right, the microorganisms and substrate, so to convert that product, the catalyst is required. Without catalyst the product it cannot be converted here. And here the catalyst screening solution development here. So because the catalyst is an essential material here, that's why, yeah, how to develop that catalyst and how to screen that particular catalyst here, because we have variety of so many catalysts are there. So from that catalyst you have to screen the catalyst and you pick up the more useful catalyst here. And next, the genetic engineering here. So the genetic engineering technique here. So by genetic engineering also we can apply this, means where you can produce some microbes here, right? You can produce some microbes by this method, and you can go with this genetic engineering, right? Genetic engineering, recombinant DNA technology, nothing but you are the genetic engineering here. Right? In that, there are two methods, sub methods are there: the recombinant DNA technology and site-directed mutagenesis here. So mutagenesis means where we can insert the different sequence of the genes. Okay, we can insert the different sequence of the gene in that particular existing gene here, and you can what is that? You can produce the different, right? You can produce the different properties or the characteristics of right, the different characteristics here. Because if any other genes are inserted, right, so then the some features will be, right, the some features will be changed here. And so for that, the useful features you can introduce. The useful features are the advantage features you can introduce by this recombinant DNA technology here. Generally, it is a genetic engineering method, and the process engineering here, and after that, here you see that every method is having its own advantages and disadvantages, right? And in the what are the advantages of this fermentation, and what are the disadvantages of these fermentations? These are nothing but the limitations here, right? The limitations. What are the limitations of this fermentation technique? You can see in this particular topic here. Now we'll see the catalyst here. The very primary, the basic requirement in this fermentation technique is the catalyst here. Okay. So here the generally in this particular fermentation technique, the catalyst nothing but your microorganism, right? The microbes like bacteria, fungus, and virus, whatever. So this acts as a catalyst here. Right? They are converting the products generally, right? Okay, and some structural features of this catalyst here. So what are the structural features of this catalyst? You see, the catalyst must be the small size here, and it is having very high surface to volume rates here. Means the catalyst size it should be small. Okay, because you see all these are the biosynthetic reactions here. All these are biosynthetic reactions. So if that, so you see in the biosynthetic reactions where you see that enzymes are involving in that. Okay, so if the catalyst are small size, then it is very right. So rate of reaction will be more here. Okay, because the non-involvement in the reaction, right, is there, and where it is having a high surface to volume rates here, right? Where the catalyst, because that's in the right here the what is it, biosynthetic reactions here, So generally the reactions are takes place on the surface of catalyst here, on the surface of catalyst here. So if the surface area, right, the surface area of catalyst is more, then more number of reactions takes place and more products are obtained here. More products are formed here. That's why these features must be validated here, like small size and the surface area is more high, the very high surface area, right? So then if the surface area is high, then it enables to the rapid transformation of that reactions here. Okay. Next, catalyst screening, selection, and the development here. Right. So the now the catalyst screening and the development here, because so many variety of catalysts are available. So you have to screen the catalyst and you pick up the more prompt catalyst or the more efficient or the more prompt catalyst here. So then how to screen that catalyst? You see here the sequence of that screening method. Yeah, here you see the microorganism from soil here, mud, lakes, and rivers or sludge are okay, sludge from effluent treatment plants. So here the like you see the microbes, right? The microorganisms, and you can find in right. So in all kind of areas here, everywhere, right? You can find the microbes in everywhere here. So here you see that microorganism from all sources like soil here, mud, lakes, rivers, and all from that. You collect, okay, you take all that and then you do the screening. Yeah. Screening means screening nothing but the separation here. Okay, separation. And from upon screening, yes, microbes with desirable properties. Right here, we are taken irrespective of, right. So irrespective of their properties, right? Irrespective of their properties, irrespective of their structure, irrespective of their what is that other features here, right? So we will select all the microbes here and we'll do the screening here and what are the properties we required. So that will be selected here, right? Upon screening. So the properties which are required, that the desirable properties can be picked up, right? You can pick and you will select those things and remaining are the outliers. You can remove those, right? So the and later here the inhibitor added to prevent the growth of the undesirable microbes here. So once pick up that microbe here, okay. Generally here that particular microbe is having that particular property, our desirable property here, right? So then we will stop that particular, right? The further uh, development of that microbe here, right? So to prevent that, you have to add some inhibitors like your drug inhibition like that. So you have to add the inhibitors here, right? So for the growth of the undesirable microbes, right? And then you see the suitable microorganism, which are by this method, which is picked, you selected the suitable microorganism, right? Then you go with this development and genetic manipulation. So then after that, you have to, right? You develop that genetic manipulation. Means you can change the sequence, right? The genetical information. You can change the genetical information here, because by doing by uh, inclusion or the exclusion of that some genes here. Okay, so then you can do that some right? So mutations here. Right? You can do the mutations process, and finally you will get the new mutant, right? The new mutant with enhanced and desirable performance level here. Okay. So by doing mutations means what we are doing, we are changing the sequence of the gene here, right? So some here you see so that DNA is there. Okay. Right. So in the gene, right? So here is the gene actually, you know that. Right. So this is the gene here. The gene. So on gene, you see the DNA here, right? So you the gene here, DNA. Okay. So generally here the DNA is having the sequence like AUG, right? So what is that? ATGC, for example, here, right? Like CGT. Okay, so this is a sequence. So then for example, you remove this and in place of A, you add some uh, G here like that. Then what happens here? Where it is formatted gene, right? Is a formatted gene here. So formatted gene means generally we'll see that the mutant which you are going to introduce in that you are going to change that should be having the enhanced properties here or desirable properties here and some new features are there, right? Then you will generate the new mutant here and that mutant is having that the enhanced and desirable performance level here. Okay. And by this mutation method, so we are producing right. So some products here. You see how that production is takes place. The mechanism, right? What mechanism in involving in that you can see this sequence of reactions. The central metabolism here, where the aspartic acid, right? This amino acid is taken. Okay, and by adding that ATP here, right? So where this by adding the adenosine triphosphate. So where one phosphate is added to this one, right? So then it becomes aspartyl phosphate here. Okay. Then by this, what is it? Yeah. So upon further transformation, right? Upon further transformation, it is going to form that aspartate semi-aldehyde here. And later, after that, you have to add the homoserine dehydrogenase, right? Homoserine dehydrogenase enzyme here. Then you will get the this particular L-homoserine here, right? So L-homoserine. So this L-homoserine, so then it will be, right, gives further, it is going to convert it, transformed into the L-methionine and L-threonine here, right? The further transformation. And this L-threonine by feedback inhibition method, again, it produces the same, right? It produces same aspartyl phosphate. So it is a cyclic process here. And this is also same thing. L-lysine here, where it is going to give the same. So from variety of starting material, right? So what do you observe here? So you can produce the same molecule like aspartyl phosphate. This is an intermediate compound, right? This is the intermediate compound to produce this L-homoserine here. So this intermediate compound is produced by, right? Produced by these all these intermediate compounds like L-lysine and this what is it? L-threonine here. Okay? All right. So next here the genetic engineering and recombinant DNA technology. Here the recombinant DNA technology and genetic engineering. So we'll discuss tomorrow. So actually, yeah. some. Okay, so I'm stopping here. So we'll discuss remaining topic tomorrow.